everyone, and welcome to our webinar, Achieve Optimal DNA Extraction from Inhibitor-Rich Environmental Samples. My name is Claire McLean, and I will be your moderator for this presentation. We will be joined today by Nelson Kottram, a field application scientist here at Omega Bio-tek, and Dr. Kiran Durvasula, one of our product managers. On the agenda for today, I will first be giving you an overview of our company and introducing our speakers. We will then explore our DNA extraction strategies for environmental samples, as well as our automation capabilities and large volume eDNA applications with Nelson Kottram. From there, Dr. Kiran Durvasula will be showcasing our product solutions for different environmental applications. At the end, I will be moderating our live Q&A session with questions submitted using the Q&A tool. As I mentioned earlier, I am Claire McLean, a marketing associate here at Omega Bio-tek, and I will be your moderator for today's webinar, which will be presented by Dr. Kiran Durvasula and Nelson Kottram. Nelson Kottram is a field application scientist at Omega Bio-tek. He obtained his degrees at the University of Sao Paulo and the Federal University of Rio de Janeiro. His subject of study during his master's degree focused on human genetic diseases and population genetics. While working as an R&D scientist, he developed point-of-care diagnostic devices based on nucleic acid amplification and single-molecule fluorescence techniques. At the Institute of Medical Genetics in Wales, he analyzed the results of NGS and Sanger sequencing assays for the diagnosis of human genetic diseases. As part of our FAS team at Omega Bio-tek, he is responsible for providing technical support for our European customers and distributors, as well as installing and validating nucleic acid purification solutions on high-throughput automated platforms. Dr. Kiran Durvasula is a product manager at Omega Bio-tek, where she leads efforts to develop and deliver novel nucleic acid isolation products for various research and diagnostic fields. She obtained her PhD in chemical engineering from the University of Florida, Gainesville, and went on to receive postdoctoral training at the Georgia Institute of Technology. From there, she served as a biological scientist at Open Cell Technologies Incorporated, developing an innovative intracellular delivery and transfection device before joining the team here at Omega Bio-tek. She has authored several peer-reviewed journal articles and is passionate about translating fundamental research platforms into practical technologies. Our vision here at Omega Bio-tek is to be the leading global provider of high-quality nucleic acid purification solutions. We set out to do this with our mission to continue to innovate while providing an unparalleled customer experience. This can be seen by our success in delivering a simple, straightforward business process built on integrity that affords our customers the best solutions on the market. We offer over 900 products across a myriad of sample types and formats, which enables us to serve an unrivaled variety of end markets and applications. Our purification kits are available in 3 formats: plates, spin columns, and magnetic beads. Our primary applications span molecular diagnostics, drug discovery and development, academic research, biobanking, as well as the agriculture, fishery, and environmental industries. Working with Omega Bio-tek provides the customer with a range of advantages. Better performance from our products includes increased downstream sensitivity and faster performance times. Our application support offers consultation, scripting, and installation of automated methods on site by our expert application scientists. Demonstrations and automated testing are available on our in-house platforms. Our flexible product solutions and sizes offer everything from individual components to customized kits to match our customers' needs. With quality products and services delivered on time, there is an exceptional value to be gained from working with Omega Bio-tek. Our average customer sees a significant reduction in their consumable costs. We are ISO 13485 and 9001 certified for quality manufacturing that allows product lot traceability with rigorous quality and process controls. With that, I will be handing it over to Nelson Kottram to discuss our DNA extraction strategies for environmental samples.
So thank you, Claire, for the introduction. I'm going to talk about the extraction of DNA from environmental samples, the challenges one would face, and Omega Bio-tek's solution to overcome these challenges. Next slide. Before I get into the DNA extraction strategies, I want to give you a big picture about the scope and applications using environmental samples. Environmental samples can be collected from a multitude of sources such as sediments, soil, surface swabs, water, animal products, stool, plant remains, and others. Once the samples are collected, the next step is to extract the DNA from all types of organisms containing these samples. The extracted DNA can be used in a variety of downstream processes, such as PCRs and sequencing. These will allow for the identification of each of the species present in these samples, allowing for taxonomic classification based on the known sequences and biodiversity surveys. With advances in next-generation sequencing technologies, it will be possible to derive information and analyze entire communities simultaneously from a single standardized sample. The potential for using environmental samples is immense. Applications include water quality management and pathogen detection in public health monitoring and surveillance by identifying transmission sources and even predict a possible outbreak, or in metagenomic analysis and microbial diversity for ecological studies, among others. Next slide. In spite of great promise, the full potential of utilizing environmental samples in the applications discussed in the previous slide is still at a nascent stage and yet to be fully realized. Some of the major challenges are: 1, getting a representative environmental sample so that we can avoid selection bias and that the data reflects the actual characteristics of the sample. 2, to get a representative sample, the input volumes are usually large, which represents a technical challenge in terms of reliable DNA extraction strategies that can accommodate variable sample input, as well as allow for high throughput processing. Number 3, these samples usually have a large quantity of inhibitors, such as humic acid and other organic matter that can co-purify of DNA that can negatively affect downstream analysis, such as PCR and next-generation sequencing. Number 4, the identification of the species is based on genetic reference databases, and since this field of study is still under development, there's lack of reference for the results. Next slide. To overcome the DNA extraction challenges and provide inhibitor-free DNA from environmental samples, Omega Bio-tek has developed the MacBiome Environmental DNA 96 kit. This kit can be used as several different environmental sample types, such as soil, water, and others, and can extract DNA from microorganisms such as gram-positive and gram-negative bacteria, yeast, fungi, and any environmental DNA that may be present, such as human, animal, and plant. This kit is magnetic bead-based and can work with variable sample input amounts, so the protocol is scalable, automation-friendly, and very versatile. This kit does not use organic buffers, allowing for safer handling. It also includes specialized proprietary buffers for the removal of inhibitors. Next slide. On this slide, I'm going to give you an overview of the protocol for the MacBiome Environmental DNA 96 kit. After collection, if necessary, samples are pre-processed according to their nature. They're liquid, solids, or salts. Pre-processing steps could include centrifugation or filtration of a membrane to concentrate our sample of interest. Samples are then homogenized, usually using bead beating. This kit includes a disruptor plate, which has pre-highly coated large and small ceramic and glass beads for efficient sample and cell membrane disruption. The samples are then subjected to chemical and enzymatic lysis, and after the lysis, we perform the inhibitor removal step, which is followed by the binding of the DNA to the beads, washing, and elution of the DNA. The eluted DNA is now inhibitor-free and ready for the downstream application of choice. Next slide. Now, I'm going to walk you through the product performance data that we have generated internally. We carried out experiments on both soil samples as well as water samples, measuring the results against comparable column-based kit from a competitor. We used 250 milligrams of soil spiked with a gram-positive bacteria and 100 milliliters of water spiked with both a gram-positive and a gram-negative bacteria. The yield and purity were assessed using a nanodrop, and a qPCR assay was performed to check for the presence of inhibitors. The results for the test of the soil samples show that the MacBiome environmental kit produced over 2 times the yield of those obtained by the competitor kit. Both the 260-80 and 260-30 ratios were also higher than those for the competitor kit. The PCR analysis comparing a 10 times dilution versus the undiluted sample resulted in a delta CT of around 3.3 between the diluted and undiluted eluates, indicative of inhibition-free amplification. There was no detectable amplification in the undiluted DNA extracted using the kit from the competitor, suggesting inhibitor carryover from the sample to the eluted DNA. In contrast, DNA purifying using the MagBiome environmental kit exhibited good amplification potential at undiluted levels, corroborating the efficacy of its proprietary inhibitor removal reagent employed during the extraction process. The results for the test with the water samples show that the MagBiome environmental kit produced higher yields than those obtained by the competitor kit. The 260-80 ratios were close to the theoretical 1.8, and the 260-30 ratios were higher than those obtained with the competitor kit. The qPCR analysis of 10 times and 100 times diluted eluates produced lower CTs than those observed for the competitor kit, indicating higher sensitivity. And the delta CT of around 3.3 between the dilutions again indicate inhibitor-free amplification. The results from both soil and water samples indicate that the MagBiome environmental kit can produce not only better yield and higher purity, but also was more effective at removing the inhibitors. As mentioned previously, this kit can be automated using a variety of automation platforms for increased ease of use or high throughput processing. The MagBiome environmental kit can be automated on most open-ended platforms, such as Kingfisher magnetic processors and Hamilton antique and liquid handlers, among others. Besides having ready-to-load scripts for the most common platforms, we also provide application support to help develop customized protocols for different samples, sample sizes, and workflows according to your needs. These are the processing times for some of the platforms we support. So on the Kingfisher, BioSprit, and MagMax, 96 samples can be processed in about 14 minutes. And on the Hamilton Microlab Star with the recommended Omega Bio-tek configuration, 96 samples can be processed in around 1 hour, and 384 samples can be processed in around an hour and a half. So now I'm going to introduce my colleague Kieran, who's going to talk about the case studies and applications.
Thank you, Nelson. Hello, everybody. My name is Kieran Durvasula, and I will be the presenter for the second half of this webinar. I will walk you all through 3 case studies that I've selected from literature that uses environmental samples as inputs and uses Omega Bio-tek's kits to extract nucleic acids from those samples. So this is basically an exercise to give you an example of how these environmental samples are used in real-life scenarios. So without further ado, let's move on to our first case study. So the first case study is going to be on phytoremediation and microbiome characterization. The sample type is soil here. To those of you who are not aware of what phytoremediation means, it's nothing but treatment of pollutants and contaminants from groundwater or soil through use of green plants. Due to rapid industrialization and rapid expansion, there's a lot of heavy metal contamination in groundwater and soil. Heavy metals such as mercury and cadmium often feature in soil, and they're often detrimental to human health and entire ecosystems in general. So there's a break in the policy at country level because there are no guidelines on how to treat these. And in the sense, mercury still features in gold mining operations and cadmium still features in use of fertilizers and pesticides that are used in agriculture. So the need of the hour to have a methodology to remove these heavy metals from polluted ecosystems, because these heavy metals are non-biodegradable. The study objective is to evaluate the phytoremediation process in the presence of perennial ryegrass in symbiosis with a fungi, mycorrhizae, and look at the uptake of these heavy metals from contaminated ground soil. And the authors also looked into the changes of microbiome in the presence and absence of the fungi mycorrhizae. So the methodology that they followed was they used our MagBiome environmental DNA kit, which is the star of this webinar, to extract DNA from contaminated soil. They followed it up with a 2-step PCR and amplified the 16S rDNA region of the microbiome and also use pair-ended sequencing to look at the microbiome characterization. So the results are presented on the next slide. I'm not going to go too much into detail, but if you look on the pictorial on the left, The application of mycorrhizae in symbiosis with the perennial ryegrass, which is Lolium perinae, definitely increased the uptake of heavy metals such as mercury and cadmium from the soil. And it also stimulated the bioaccumulation of these heavy metals in the plant itself. And on the right, the DNA was put through pair-ended sequencing. It was really, high quality. And the stacked bar chart there gives you an idea of the relative abundance of bacterial families that are present in the soil for different treatments and replicas. And, the main takeaway there is basically in the presence of mycorrhizae, the communities were distributed such that they contributed to increase in the response of the plant to stress. So in conclusion, the paper basically suggests that mycorrhizae in symbiosis with the Lolium perinae is a good alternative to just using the ryegrass as a sustainable technique to remove heavy metals from the soil. And that's a good method to control soil pollution. So the second case study, I would like to switch gears a bit here and talk more about the public health, surveillance, and monitoring aspect. And till now, we've been talking about DNA. Given the recent COVID pandemic, there was a lot of focus on viral RNA from environmental samples. So since the topic is environmental sample, I just want to give you a little flavor of RNA from environmental samples and how Omega Bio-tek has kits that can actually extract viral RNA from environmental samples as well. So, given the recent pandemic, there was a lot of focus on wastewater as a promising source for surveillance and to looking at the spread of SARS-CoV-2. Typically, composite sampling is what is used for doing this sort of a work. However, composite sampling uses autosampler, which is expensive, and it's often limited because of intermittent flow issues. So to be able to fully exploit the potential of using wastewater, the need of the hour is finding an inexpensive and easier to implement sampling technique that can actually be utilized in a broader implementation scenario. Here, the authors looked at two wastewater collection methodologies. One is the commonly used composite sampling with an automatic sampler. And they also looked at passive sampling using more swabs to look at the viral detection and the viral load from these two sampling technologies. So the RNA was extracted using our MADBIND viral DNA and RNA 96 kit, which was followed by a one-step differential display RT-qPCR, which used a combination of primers to look at different variants of SARS-CoV-2. And they also looked at tiled amplicon sequencing to be able to sequence this variants of SARS-CoV-2 RNA genome. Moving on to the next slide. So the left is a pictorial of, the experimental plan. This was carried out on a college campus. So they had like 4 different manholes where they grabbed samples from. And the main takeaways are, the Moore swab, which is a passive sampling, inexpensive technique, actually did better in terms of, RNA quantification in terms of copies per mil of wastewater that was sampled as compared to composite sampling. That's the graph on the top right. And the one in red is a Moore swab. And in terms of variant identification through whole genome sequencing, both the sampling technologies were comparable. Another interesting, finding from this paper is since they were continuously monitoring, they were actually able to see the introduction of the new variant into a monitored community. So definitely interesting paper. I do recommend you all check out. The reference is at the bottom of the slide. And again, the implications are immense. This kind of leads pathway into using, passive sampling technologies for, wastewater sampling and overall, surveillance of emerging pathogens from wastewater. The third case study is on public health transmission and control strategies. This is another interesting topic. A lot of parents in the audience might be interested to, actually learn more about this. Parents often worry, especially during the Halloween time, the risk of transmission by, due to trick-or-treating. So this is what the paper actually looked into. It looked into, what is the risk of SARS-CoV-2 transmission through these candy wrappers? Again, very, very interesting topic. So they tried to quantify this risk. And as a study objective, what they looked into was like, what if the candy giver washes their hands, and what if the recipient, after getting the candy, actually washes the candy wrappers before opening and actually consuming the candy. So they kind of looked into that and looked at the viral load before hand washing by the candy giver and after washing the candy by the recipient. Again, the RNA from, so they basically used the swab to, swab the wrapper surface and use that to extract RNA from the swab. And they put it through an RT-qPCR using a thermostat bath multiplex assay. And they also looked into an inexpensive loop-mediated isothermal amplification, also known as LAMP, to check how well it matches the RT-qPCR results. The next slide. So this is their experimental strategy. They looked at 10 independent individuals where the candy giver either washed the hands, didn't wash the hands, or they coughed on the candy. And after receiving the candy was either put through, either not treated at all, or washed with a household detergent for little over a minute, and then all these groups were put through RNA extraction and the RT-PCR and LAMP that I had discussed. So the graph on the right, I'm not going to go into too much of a detail, but what it essentially tells us is hand washing prior to handing out the candy actually greatly reduced the positives. Like if you look at the group that is black on the right side of the graph, which is the washed group, you see there's a lot of those black rectangles. So those are all COVID negative. And similarly, after receiving the candy, washing the candy in a household detergent also reduced the viral load significantly. So the main conclusion here that we can draw is by taking these sort of precautions, the transmission of SARS-CoV-2 is negligible. However, COVID is primarily an airborne, transmitted disease. So precautions such as mask wearing, social distancing is, recommended, however. So this is the last topic I'm going to talk about today. It's a large volume eDNA application. We worked at a customer site and placed a solution there. And this is another hot topic in the environmental area, which is the environmental DNA. Again, I just want to go over a few basics of environmental DNA on my next slide. The first question that I would like to answer is, what is eDNA? So eDNA stands for environmental DNA. So it's basically the DNA that is shed by organisms into environment. So this could be basically hair, feces, skin cells, secretions, etc. This is often found in soil, sediment, biofilm, as well as water. So the sample types here are those that are listed on the left there. And this is actually, gaining a good bit of traction because as Nelson actually pointed out before, what it allows us to do is actually get a community level idea and look at multiple species than just single species. And again, the sampling is very efficient, non-invasive, and very easy to standardize. And the DNA, once extracted from those sample types, can give us information about spatial, temporal, biodiversity patterns. You learn about taxonomic groups and essentially the biodiversity. On the next slide, I just want to, give you all an idea about the solution we placed at a customer site. Typically with environmental samples, the volumes are a lot because you want to get like a good representation of all the organisms that might be present in that sample. So in this case, it was actually marine sediment and the input was like 10 grams. So those of you who work in this area understand the challenge of processing a sample as big as that. Another big challenge that we had to deal with was the customer wanted to move on to automated processing. So we helped them transition this solution into an automated workflow from their existing manual method. Sample disruption and inhibitor removal buffer was used to be able to one, efficiently homogenize the sample, and two, eliminate those PCR inhibiting compounds that are often present in samples such as these. So we were able to obtain high quality DNA that was later on used in metabarcoding studies. And the customer was actually able to derive powerful biodiversity information from that sample. With that, I would like to conclude by saying that our MagBind environmental DNA 96 kit can extract high quality DNA from environmental samples, eliminating the PCR inhibitors that are often present in samples such as these. And in fact, the data that we showed earlier, in most cases, we were actually able to get better quality with low level of inhibition compared to our competitor. The kit follows magnetic bead-based approach, making it automation and high throughput friendly, as well as, it can be scaled up. I'm hoping that with the 3 case studies that I discussed, I brought home the point that we have a wide range of kits that can be used for like different applications. Given the nucleic acid that you would like to extract from your environmental samples, there may be like one kit better than the other. So with that, do connect with us. We are always open to talking with you all. Do discuss your project application, and we're always there to help and make recommendations. So you can reach us at info@omegabiotech.com. And I would also encourage you all to utilize our free product sample program that we typically carry out. Go to our product page. There should be a link for requesting a free sample. You get to try out our product before you actually make the decision to buy it. With that, I thank you all for your attention and pass it back to Claire for the last few words.
Thanks, Kieran. I would like to thank everyone again for being here with us today. We will be using our last few minutes to answer some questions from the audience. If you have anything you would like to ask our presenters, please submit it using the Q&A button located on the toolbar. Let's see. Our first question is, can I process different sample types than soil, stool, and water with the MagBind environmental DNA kit?
Yeah, I can take that question. Thank you everybody for attending our webinar. Like we've presented in the webinar, if you look at the pictorial for the eDNA, we did mention a lot more sample types like biofilm sediment. So yes, this kit can definitely be used for more than one sample type. However, the pre-processing steps could vary. So do connect with our team so that we can give you the right product recommendation and protocol recommendation.
Awesome. Thanks, Kieran. If I wanted to automate my eDNA purification with the MagBind environmental kit, how should I start that process?
Okay, so if you want to automate the MagBind environmental kit or any other of our kits, just get in touch with your sales representative or our automation advisor. You can find this on the website. And we were going to work with you to develop the best solution to fit your needs and workflow, and advise on instrument, work with potentially instruments that you have already. And we can provide support all the way from the initial consultation, so regarding what's the best kit, all the way to the implementation of the method in your laboratory.
Great. Thanks, Nelson. I'm having difficulty lysing gram-positive bacteria from my water sample with the environmental kit, and it's causing my DNA yields to be low. What can I do to fix this?
Yes, yeah, the gram positives can be tricky sometimes. So in our experience too, when we use filtration technique to, concentrate the sample, it's hard to get those microorganisms off the filter to be able to efficiently lyse it. So there are some tips and tricks behind it. One, you could do like an enzymatic lysis on top of bead beating. And sometimes adding a detergent when we do the bead beating actually helps to increase the gram-positive lysis.
Thanks, Kieran.
Mm-hmm.
Are Stereofx filters compatible with the environmental kit? And what other filters are compatible?
Stereofx filters are compatible with the MacBio environmental kit. And in fact, we have actually developed a method to lyse the sample without having to remove the filter from the cartridge. And that can reduce the amount of manual handling as well. So there's no need to break open and manually remove the filter. Other than the Stereofx filters, we also have the kit is also compatible with polyether sulfone filters.
So what inhibitors can't be removed by your inhibitor removal buffer?
Yeah, that's a good question. Honestly, we've not done those kind of studies to look at what are all the inhibitors that are found in the samples and what we are actually removing. The commonly found inhibitors such as the fulvic acid, humic acid, we are able to remove, but this is a question I can't really give you a comprehensive answer because we've not really done those kind of studies. Yeah, but from the data, it's apparent that we are able to remove most of the inhibitors that are found in the samples that we have tested.
What is the average quality of microbial metagenomic DNA extracted after bead beating in terms of shearing? Is the average fragment size 1 kb, 10 kb, 100 kb?
I can answer that. Well,
okay.
Kieran?
Yeah, go ahead, Nelson.
Yeah.
Yeah.
It's basically we would have to do a test and optimize the bead beating so there's enough bead beating to shear to break up the cells without shearing the DNA too much, and also being representative of the environment you have, of all the community you have. But with bead beating, we're looking more towards 10 to 30 kb rather than 100 kb.
All right. And our final question, can this tool find paleo-environmental organisms and their characteristics?
So the extraction kit can give you the DNA from the environmental sample, but the downstream is what is more important and to be able to find those organisms, if that makes sense. Because one, it depends on if those organisms are already existing in the reference database, right? Because a lot of the sequencing is limited based on the database restrictions. But the extraction part, for sure, our kit is and can extract comprehensive DNA from the environmental sample. And studies such as these, you do have to optimize the sample input as well, because sometimes if you start with too small a sample, that may not be like a comprehensive representation of the organisms that might be present. So in short, yes, but extraction-wise, yes, but in terms of characteristics and actually assigning a taxonomy to those organisms, that's dependent on the downstream.
All right. Thanks for your answer, Kieran. That does it for our questions for today. Thank you everyone for being here. If you would like to request a sample of any of the kits discussed here today, please visit omegabiotech.com or get in touch with your sales representative. We hope to hear from you soon.
Yeah, thanks, everybody.
Thank you.